All right, it's just after past. Good evening, everybody, ladies and gentlemen. My name is Tanya Reinhardt, and I'm the Science Center Coordinator at the University of KwaZulu-Natal Science and Technology Education Center. And on behalf of the University of KwaZulu-Natal's College of Agriculture, Engineering and Science, I would like to welcome you to our second online talk in celebration of National Science Week. This talk is the continuation, actually, of our What Do Scientists Do? A Look Inside the Ivory Tower public lecture series. And before I introduce our guest speaker for this evening, just some house rules, please. Please make sure that you have muted your microphone. If you don't know where it is, at the bottom left of your screen, you have your little console, and there is a little microphone. So please mute yourself, otherwise we mute you. Also, please, it's going to be a difficult night in terms of bandwidth, so please switch off your camera just for us to be able to screen. We will have a question and answer session afterwards, and you can post the question in the chat, or you can just unmute yourself, or raise your hands and we unmute you in order to give the talk. To ask the question. Okay, just a brief background on National Science Week. So National Science Week is an annual celebration of science, engineering, and technology, attracting thousands of learners and members of the public to workshops, science shows, and lectures, which are held at universities, schools, science centers, and public facilities countrywide. The Department of Science and Innovation leads the initiative, and the South African Agency for Science and Technology Advancement, NRF-SASTA, is responsible for its implementation. Each year there is a different theme, and activities usually revolve around this particular theme. This year we are, and I think it's really a nice topic, celebrating the role of basic sciences in the modern world. And tonight we are celebrating the achievement of a young researcher, Ms. Zakithi Mkhize. Let me just quickly introduce her briefly. Zakithi is a PhD candidate at the HIV Pathogenesis Program, HPP, at the Nelson Mandela School of Medicine at the University of KwaZulu-Natal in South Africa. She's also a Sub-Saharan African Network for HIV TB Research Excellent, SANTHE, PhD fellow. And I know this is like quite a mouthful. So you see, I'm trying to breathe in between. She's currently working on HIV cure studies, and from yesterday we saw that this is something still burning, particularly on understanding the dynamics and transcription during HIV infection and latency. Her project focuses on investigating how genetic variation in HIV regulatory protein T8 plays a role in interacting with host transcription factor, positive elongation factor B, and again, some weird abbreviations, and you have to explain it to me later on, PTEFB. She is passionate about using science to make life-changing medical discoveries and is hopeful that we will see a cure for HIV in our lifetime. And I think that everybody, I think, wholeheartedly, wishes you good luck with that. Zakithi is also an avid science communicator and youth STEM activist. Her YouTube channel, Black Girl Scientist, is used as a platform where she shares her journey through science, helping others just like her to navigate the field. She says that COVID-19 pandemic, although devastating in many ways, helped to highlight the important role which scientists play in the world through doing basic science. I would now like to leave the microphone to Ms. Zakithi Mkhize and her talk, Doing HIV Research in the Lab, the Hunt for the Cure continues. Zakithi, it's your platform now.
Thank you so much. Thank you so much, Tanya, for that introduction, and good afternoon to everybody. I am just going to take a moment to share my slides. Okay, I assume everyone can see the slides. Can you just confirm for me?
Yes, we can.
Yes.
Thanks, Zakithi.
Yes.
Thank you. Thank you. So good evening, everybody. Thank you so much to the University of KwaZulu-Natal, as well as ARI, also the Department of Science and Innovation, and all the other partners in really bringing this public online lecture series. I think it is really important. And although it seems like things are kind of going back to normal after the most devastating times, I think during the COVID-19 pandemic, hopefully we'll be able to have more in-person interactions. But I'm very excited to be here, and thank you for the invitation. So yes, as Tanya said, I am going to be giving a talk today, and the title of my talk is Doing HIV Research in the Lab, the Hunt for the HIV Cure Continues. So before I get into my talk, just a little bit about myself. I was born and raised in Durban, and I think for me, science, my passion for science was very intrinsic. It was, well, at least I can't pinpoint exactly when it was, or something that kind of sparked the interest in science subjects for me. But I remember during my high school years, I was very interested in what we had, life sciences and physical sciences. And I also did well in those subjects. So I think that's what really ignited my passion for science. And of course, then I enrolled at the University of KwaZulu-Natal to do my Bachelor of Science, in which I majored in genetics and microbiology. Following that, I did my honors in microbiology. And then, you know, kind of in following, I know when I was growing up, I didn't really know that I wanted to be a scientist. I don't think that that was a career that people a few years ago would grow up and think that they want to be. I don't think I even knew what a scientist was back then. It was back then you're either going to be a lawyer or a doctor, a teacher, an engineer. I don't think scientist was something that was at the top of very young people's list. And I think that's because people don't really understand what it is that scientists actually do. So I was really interested in more of the medical research, and testament to that, when I enrolled at the HIV pathogenesis program to pursue my master's. My master's, which I achieved in 2018 in virology. I achieved my master's cum laude in that year. So also with HIV research, of course, if you are South African and you live in South Africa, you are either infected or affected by HIV. So that was another driving force in me wanting to pursue, you know, in being a scientist that does HIV research. I really wanted to understand the virus better and also do my bit in contributing to finding an HIV cure. I remember when I told my mom for the first time that I was going to do HIV research, she told me many stories about how in the 90s, you know, in Black communities, a lot of people were dying. And she described it as you kind of just see the life being sucked out of people when they become infected with HIV. And back then, people became infected with HIV and very quickly progressed to having AIDS. So yeah, that was really, really, you know, another driving factor, which pushed me to pursuing, you know, being involved in HIV cure research. And of course, as mentioned in my intro, I'm also very, very passionate about science communication. I have been in the top 10 National for Fame Lab, which is a very, very popular science competition. And I've also, it's kind of intriguing for me, for as long as science has been around, no one has ever thought that science communication until recent years is important. For me, that's just mind-boggling about how scientists would think that they would do science for people, but they're not communicated to those very people that they're doing science for. So, you know, being a science communicator is something that's really, really close to my heart as well. And COVID-19, again, as said in my intro, although it had many devastating effects, it really played an important role in highlighting the importance of science and the role that basic sciences play in modern life. And in science communication, I think we also saw during the COVID-19 pandemic, which is a classic example of the mistrust between scientists and people. We saw a lot of vaccine hesitancy, etc. Yeah, so I'm also, as a young person who does science, I'm very passionate also about, you know, I've been attending a couple of National Science Week engagements, and in my previous engagement, a speaker there said, you know, which I thought was a golden nugget, that it's really important to lift people with you as you rise. So for me, empowering young people who are also pursuing careers or going through the journey of STEM, which is science, technology, engineering, and mathematics, is really important. And how can I make their journey easier than mine? How can I make it better than mine, so that their road to success is really seamless? So yeah, that was a little bit about me. And of course, the reason why we're here is because of National Science Week. As mentioned, is the theme for this year is celebrating basic sciences in the modern world. National Science Week launched on the 30th of July, and there have been many engagements, very exciting, and it's beautiful to see, you know, how people are getting really excited about science and getting young people excited about science as well. So when they grow up, you know, maybe a couple of them will say, I want to be a scientist, you know, when they grow up. So the objectives of this National Science Week, and I've highlighted kind of the words that really summarize these objectives. And the first one is to illustrate. So it's to show the role that science has in tackling problems and challenges encountered in creating and sustaining a prosperous society. The second one is to demystify the myths surrounding some of the innovations. And again, with COVID-19 being a classic example of how we've had to demystify myths about, you know, the vaccine and 5G, etc. With the people having vaccine, there's been a lot of vaccine hesitancy. The third objective was to display, to show South Africa as the home of some of the discoveries and innovations that influence the world. And then finally, to raise awareness of these STEM careers. So what are basic sciences? I think yesterday also, Tanya, who introduced me, said, you know, she doesn't think these sciences are basic. And I agree. You know, basic sciences are really, in my opinion, the fundamental building blocks of life. And these scientific disciplines of basic sciences are mathematics, physics, chemistry, and biology. And they provide fundamental understanding of natural phenomena and the processes by which these resources are transformed. So in me saying, you know, that they are the building blocks of life, it's like, you know, imagine a house. When you build a house, if the foundation is not stable, the house is not going to really stand and last very long. So basic sciences are really, you know, the fundamental building blocks of all these science subjects. And without basic sciences, we really wouldn't know, for example, that SARS-CoV-2 causes the COVID-19 disease, and we wouldn't know that HIV causes AIDS. So really, in short, these sciences help us to fight pandemics and its consequences. And all of these things are rooted in basic sciences. So this was just a figure that I found, which I thought is really important in highlighting how basic sciences enable many different disciplines. You know, the banking and finance sector, IT, we hear on this virtual platform this afternoon because of science, you know, engineering and mining, and the list really goes on and on in the role that these basic sciences play in those sectors. So yeah, we've been speaking a lot about science, and it is actually scientists that do science. So what exactly that we do is that we conduct and gather research to further knowledge in a particular area. Typically, scientists will make hypotheses. So it's to really answer questions that are unanswered. For example, an hypothesis is basically an inference that we make. So for example, the sky is blue because it is a reflection of the ocean. And then we design experiments to really prove that. So there's several different types of scientists, and nearly every industry, which I showed in the previous slide, requires scientists in those various fields. Scientists aim to find measurable data through various analysis and testing methods as well. So getting down to the crux of my presentation, and the reason why most of you are here and joined this afternoon, and that is to talk about HIV research and doing HIV research in the lab as the hunt for the HIV cure continues. So I'll first be taking you through the history of HIV. I'll then go through different strides which have been taken in fighting HIV. And the all-important question of why there is no cure for HIV. And then I'll take you through a little bit about what the HIV pathogenesis program does, the research that is conducted there, and then I'll also tell you a little bit about my project in the simplest way possible. So first starting with the history of HIV. So it was in the early 1980s when a new and unusual diagnostic pattern began to emerge in different parts of the world. And a benign cancer called Kaposi sarcoma was common in the elderly. However, it started appearing as a virulent strain in younger adults. So at the same time, an aggressive form of pneumonia began to come up with alarming frequency in another group of patients. And in the US, in the 1980s, this kind of unusual medical problem that they had was occurring in a lot of males, gay people particularly, and it was actually called the gay disease in the US in the 1980s. So by 1981, scientists had begun to connect the dots between these new diagnoses, plus a number of other opportunistic infections. And by the end of the year, acquired immunodeficiency syndrome, which is AIDS, which is caused by HIV, was documented. So of course, I cannot speak about the history of HIV without speaking about the scientists which actually discovered this virus. So it was actually interesting in my doing research for this talk, because I learned, and I've written in my proposals, that HIV was discovered by the Barry Sonosis, which is the female here in this image on the left. However, we can't talk about the discovery of HIV without mentioning two other important scientists, which was Luc Montagnier and Robert Gallo. So they are pictured in the image on the right, where they were receiving a Nobel Prize for discovering HIV as the causative agent of AIDS. So coming back to HIV in South Africa, where do we stand and what do the stats look like in South Africa? So Africa as a whole is disproportionately affected by HIV, and actually sub-Saharan Africa accounts for 70% of the global population. So of everybody in the world that is infected with HIV on our continent, 70% of those people reside on our continent. In South Africa in particular, the adult prevalence is about 19.1%. So that's about 1 in 4 adults which are infected with HIV. And there were 230,000 new infections which were reported in the year 2020. So this is from the UNAIDS report. With regards to the death rate, which is shown in the image on the right, of the death rate of AIDS in 2019, again, you can see, our continent is very red in comparison to the rest of the country, where sub-Saharan Africa is greatly affected by this disease. So why is HIV, such a unique virus? In everything that has happened with COVID-19, and as Dr. Cele described to us yesterday, kind of from when in March 2020, when lockdowns began to happen and everything that he was doing in the lab. And a question that I think a lot of HIV researchers get is that, why is there no cure or vaccine for HIV, whereas COVID-19, within less than 2 years, there was already a vaccine. And this is because this virus is very unique. in the way in which it infects a person's body. So firstly, HIV takes over the host. So the host in this case would be the person which is infected with HIV. This virus attacks the person's immune system. So in our bodies, if you're uninfected, of course, your body is made up of many healthy cells, mostly CD4 T cells. So what happens when a person becomes infected is that the virus will bind to receptors on your CD4 cells. So these are like little feelers on the outside of your of this particular cell, and the virus itself has its own feelers. So they kind of fit together like puzzle pieces. And upon it binding to these receptors on the CD4 cell, it injects its genomic material into the cell. And then this material actually integrates into the cell's DNA. So it doesn't just infect the cell and kind of swim around in the inside of the cell and makes that cell sick. It actually integrates into the host's DNA. So as we know, as humans, we're made up of DNA, we're made up of 23 chromosomes. So basically, once this virus has integrated into the DNA of the cell, it instructs the cell to make more copies of itself. So then this cell will begin to make kind of all the pieces that will fit together to make a new viral particle. These will then assemble together and then bud off from this cell. So this cell has gone through an HIV life cycle of where it's been infected, the genomic material has been integrated, the cell has now, the host cell machinery has now been taken over by this virus, and this cell begins to make new pieces, which will make up a new virus, which will bud off from the cell and then go on and infect other healthy cells. So that is how, the infection spreads throughout the body. However, so when a person becomes infected with HIV, in some of these cells, the virus lies latent. We call it latency, which basically means that the cell is kind of asleep. So after infection and integration, there's no active replication. So the cell does not make new components of a new virus. However, the cell remains infected, and that remains a major barrier to finding a cure for HIV. So although HIV has been very devastating, I think, and it continues to be, there have been efforts which have been made to fight HIV over the years, since its discovery in 1981, right up until, I think, 2020, this timeline goes into. Just want to get a pointer so I can focus on particular years. So such as in 1994, it was approved that pregnant women could be given a particular antiretroviral drug to reduce transmission from mother to child. And also, I can't remember which year it was, but now people who test for HIV immediately go on treatment. There's no longer a wait for the CD4 count, which is all the healthy cells in the body. There's no longer a wait for those to drop to below 200, which is actually the clinical characteristics of AIDS. So AIDS is an advanced stage of HIV. And then also in 2009, we had the first patient which was supposedly cured, known as the Berlin patient from HIV, which I'll get into a little bit later. And, there've been several advances which have been made with regards to treatment. So when antiretroviral therapy first came on the scene, it was a multiple tablets or pills which an infected person would have to take. And now these have all been combined to one pill, where we can have people taking one pill a day. So because we're still kind of playing this game of hide and seek with the virus and really hunting for the cure. Another question that we get a lot is, why is there no cure for HIV? So as I explained in my previous slides, that HIV is unlike any other virus. It's an integrating virus. So it integrates into the genome of its host. And of course, within the body, because we have several cell types, it integrates into all of these cells. So these cells then hide in, our lymph nodes, in the liver, in the spleen, and that's where they lay what we call latent or hiding. So as long as a person is on therapy, they'll be able to control this replication which occurs, and they can and their viral load can actually drop to even below the limit of detection. However, when a person stops taking treatment, or there is treatment interruption, or the person doesn't really adhere very well to treatment, there's a viral rebound which occurs. So these this means basically that a person's viral load starts to increase again above the limit of detection, and there's active replication, which is now happening in the body of this infected person. And the source of that viral rebound is from these latent cells. So there's certain triggers which trigger now the cells which I previously described as being infected but not having active replication, which triggers them to now begin to replicate within the body. So we really think these latent reservoirs are the source of viral rebound when treatment is interrupted. So in order to cure HIV, we really have to eliminate these viruses that hide in these cells. And there are many approaches in which we can use in eliminating this reservoir. So the first one is stem cell transplant. And this is really how the patients which have been described as being cured have really been arrived at that kind of, post interruption of therapy. There's no viral rebound that occurs in these patients. Also CRISPR-Cas gene editing as well. However, the two that I want to highlight is these strategies. The first one is called the shock and kill, and the second one is the lock and block. So with this shock and kill strategy, it basically talks to waking up this virus that is sleeping in these cells which are infected. And then in that way, the cells begin to form new viruses. So new viruses will battle from the cell, which are then targeted by antiretroviral therapy, as these drugs, antiretroviral therapy drugs, target the virus as it buds off from the cell. So that's killing those cells which are now being produced from these latently infected cells. And then in the lock and block, this speaks to actually having those cells which are latently infected, so which have been infected but are not actively replicating. This speaks to making them go into an even deeper state of latency, so an even deeper sleep, so that they cannot be reactivated even when treatment is interrupted, etc. So those are the two most important strategies that I wanted to highlight and really speak to the kind of research that is being done at the HIV pathogenesis program and is being done by myself. So I'm sure some of you have heard, if you follow HIV research, the terms functional cure and sterilizing cure. So these cures are different, and I'll explain that by using this figure. So basically, when a person becomes infected with HIV, there's a high number of HIV which is in the blood. However, when a person begins to take antiretroviral therapy, there's this steady decline in which the viral load can dip even below the limit of detection, which I explained. However, when treatment is ceased, or when the person stops taking treatment, or there's interruption, there's a viral rebound that occurs. So that is described as in treatment failure. So with the functional cure, this speaks to basically keeping a person which is infected with HIV off therapy, but not having this viral rebound that occurs. So the viral load still remains very low in that person that's infected with HIV. And in that way, it's said in science, people, or in the field generally, that people who have low viral loads, so even below the limit of detection, there's a much lower chance of transmission that way. So that is in achieving this functional cure, where a person basically remains below the limit of detection, even when they are not taking antiretroviral therapy. So a sterilizing cure speaks to completely eradicating all virus that's in a person's body. So that speaks to, reactivating the entire latent reservoir and then killing off all those cells. So in a sterilizing cure, every trace of the virus is eliminated. So that's the difference between these two terms. So although I'm more focused on HIV cure research, I cannot have this talk without speaking on HIV vaccine, of course, because that was also a question that's been on everyone's lips since COVID-19, as in how this vaccine for SARS-CoV-2 was developed so quickly, yet we have other viruses such as HIV, which has been around for over 3 decades, but there's still no cure for it. So basically, in the world of these viruses, vaccines fall into one of 3 categories. The first category is that they respond to antibodies, which are induced by the vaccine. And luckily for us, SARS-CoV-2 fell into this category. And then there are vaccines which are about 60% effective. So for example, it's the influenza vaccine falls into this category. So they provide some sort of protection against the virus, but they are not 100% perfect. And then there's a third category, which is forming an antibody to the virus, and that's not adequate to prevent infection. And unfortunately for us, HIV falls into that third category. So in order to develop an HIV vaccine, scientists need to do very sophisticated engineering to induce these T-cell effects as well as innate effects. So that's inducing the body's own immune system and antibody effects. So there are many approaches of how to do these sciences and many scientific delivery mechanisms, which target different areas of the virus and different parts of the immune system as well. So as I mentioned, that I will be talking about the Berlin patient, which was the first patient in 2009, which was said to be cured of HIV. This person underwent a stem cell transplant from a donor which had mutations in the receptor sites of the cell. So if you remember a few slides back, when I said that you have these healthy cells that have feelers on the outside and the virus itself, so HIV also has these feelers, and when it infects a cell, they fit together like puzzle pieces. So in these donors, they have a mutation which actually doesn't allow entry of the virus into these CD4 cells. These people are very rare, and less than 1% of the population actually have this particular mutation. So luckily for Timothy, which is the man that's pictured here and is called the Berlin patient, he had cancer and he received a stem cell transplant from a person who has that mutation. So he was described as having prolonged post-treatment control after treatment interruption. So this is speaking more to the functional cure, which I described. So for as long as he still was not taking therapy, there was no viral rebound that occurred. So since then, there have been 2 other people which have been cured in a similar way. And another one was the London patient, and most recently, the first woman which was cured from HIV, and that was all over the news and social media as well, as the London patient that was also cured previously was a man. So I think recently we had the first woman being cured using this way. However, I think it's really important for me to highlight that this kind of therapy is not applicable. It cannot be applied in a large scale. We can't have everyone who's infected with HIV undergoing this very invasive surgery. And also, as I mentioned, less than 1% of the population has this particular mutation. It's something that they're born with, and we will not have even nearly enough donors for all the people infected with HIV to be able to cure people in this way. It's just not feasible at all. So we continue to do HIV research and investigate other strategies in how we can cure HIV. A lot of the research that has been done, that is being done now in the field, is looking at strategies to eliminate the reservoir. There are also many challenges, though, that we encounter in actually measuring this reservoir in infected people. And this is because we're not able to get from infected people, you know, into these sanctuary sites where the virus hides. And also when we measure the virus, for example, if we use PCR and try to amplify, you know, the DNA of the HIV genome, they are defective viruses within people. So although we may be detecting, you know, DNA, those viruses may be defective and not reactivatable. So as I also previously explained, another strategy which speaks to eliminating the reservoirs is shock and kill and lock and block. And it's also really important to identify several markers of disease progression. So how quickly an infected person progresses to AIDS, which is the advanced form of HIV, or how slowly they progress to AIDS as well. So in order to perform, you know, this research, we need really sophisticated labs. And I have been very privileged to have enrolled at the HIV pathogenesis program. For those of you who don't know, the HIV pathogenesis program is located at the Nelson R. Mandela School of Medicine at the University of KwaZulu-Natal. So the campus is pictured here. And this is the beautiful K-Ruth building, and the DDMRI in which the HIV pathogenesis program is located is just behind this building. So HPP was established in 2002 in collaboration with Harvard Medical School in the United States of America, and it focuses really on exploratory pathogenesis and translational research into HIV and TB, as well as serves as a sample repository for all HPP study samples. So the work done at HPP is, you know, as the name suggests, mostly centered around HIV. There are some people who do research on HIV and TB co-infection, as well as with other comorbidities. So to date, HPP, the people that work at HPP have published over 200 manuscripts in peer-reviewed journals. And they have 7 clinic sites in KZN, one being located at the Prince Mshini Hospital in Mlazi Township in Durban. And the lab is dedicated to studying host virus interactions, antiviral immune responses, and biomedical interventions, which are applicable to resource limited settings, such as ourselves here in South Africa. So as I mentioned, the HPP serves as a sample repository also for the studies, which are conducted at HPP. There have been many. The most recent cohort which has been set up is called the FRESH cohort, or the FRESH study. And this is the females rising through education, support, and health. And this is a longitudinal study which was to identify and analyze the immune responses of infected peoples immediately after HIV diagnosis. So these women are aged 18 to 23, and they reside in the Mlazi Township of Durban. And also in my doing this research, it was quite shocking that actually almost 70% of these young women would become infected with HIV by the time they turn 23. To me, that is absolutely just mind-blowing. And we all reside in Durban. Well, I think most of the people on this call, and not very far from where all of us live, there are young women who are getting infected with HIV at an absolutely alarming rate. So just a little bit of a look inside the lab. At HPP, we have biosafety level 1 and a biosafety level 2+ lab, where we have a molecular biology lab on level 2, and we have the immunology lab on level 1. There's also what we call the core lab, or where the team in the core lab actually receives the sample. So This is what I would look like here in the figure on the left when I'm in a P2+ lab. So the personal protective equipment, or PPE, is much more than when you're in a lower biosafety level lab. And here, as shown on the right, this was actually me in the lab on my recent research visit, which was in the Netherlands in Europe. And this was just showing myself in a biosafety level 1 lab. So here I was actually not working with anything infectious, which is why I'm not dressed in the same way that I'm dressed on the left. Okay, I'm not sure what happened with these slides here, but I was showing about how we receive the samples and then how we work with the plasma. So as I mentioned before, you know, that the HIV is an integrating virus and it integrates into the genome of an infected person. However, this HIV, the virus, has a genome of its own. So this is just showing a snapshot of what the genome looks like, where it's made up of various genes. I won't go through all of them. I just wanted to highlight here the TAT gene. The TAT is a regulatory gene which plays an important role in HIV replication and transcription. So that's the translation of DNA into proteins. And particularly the TAT protein plays a really, really important role in the process of HIV replication. Previously in my master's, I worked on really characterizing this gene in primary infection. So this is the first phase of HIV infection. And I basically looked at back then the genetic variation. So the changes or the mutations which occur intrapatiently from the HPP acute infection cohort, as well as the fresh cohort, which I previously described. So basically the findings from that study was that there is this genetic variation which occurs in patients at transmission. So basically there were no 2 patients which had the exact same TAT sequence. And there were some patients which had mutations which have been previously reported to reduce the functionality of TAT. Some patients had mutations which increase the activity of TAT. So in increasing the activity of TAT, we also enhancing transcription and replication. So we would hypothesize that those patients would progress faster to AIDS than patients who don't have those mutations. We actually showed a particular mutation at position 21 of this protein, where patients who had an alanine had a significantly lower viral load than patients with a proline in that position. So this was kind of really the beginning of my journey with TAT during my master's. And I actually presented at 2 international conferences in my master's. And it was when I attended the Keystone Symposia in Canada, where I remember we had a speaker which was speaking about TAT, however, not in the way that I had read about it or in the way that I was reading about it currently for TAT. They were really explaining TAT in relation to the way that it functions, and that's dealing with host transcriptional factors. So these are these kind of helpers that we already have in the body, which TAT uses to enhance its activity. So I remember in 2018 at that conference, I was actually one of 2 Black people then. And, you know, I attended, that was kind of the first culture shock for me, seeing how much of a boys' club first the science field was, and I think still is. And also how very, it wasn't very diverse at all. So I remember, you know, finding that talk so very interesting as I was still doing my master's. And I, at the time, I obviously had aspirations of doing my PhD. And I thought, wow, we can now look at TAT in a different way, in the way that it interacts with these transcriptional factors or helpers, which we actually have in our bodies, in the cells of our bodies. So that gave birth basically to the to my PhD project, which I was very lucky to play an instrumental part in actually designing, or at least having the idea, and of course my supervisor really refining it and adding all the scientific meat to this hypothesis that I had formulated from this conference. So yeah, my research is really focused on TAT, the important role it plays, and particularly in recruiting this host transcription factor, which is called the positive transcription elongation factor B, or PTFB in short. And that plays an important role in efficient viral gene transcription. And the major aim of my project, since I had previously kind of characterized TAT in terms of genetic variation, was to basically investigate how genetic variation within TAT affects its ability to recruit PTFB, and as well as its propensity for viral latency. So just a bit about doing HIV research in the lab. I had no idea what happened to all my illustrations here, but from receiving a sample, we basically isolate the RNA from that sample, and then we perform PCR, or polymerase chain reaction. This is routinely done in our molecular biology lab, and then in this way I was able to amplify my gene of interest, which was TAT. And then also we do various other things, and we in the second part, which is what we do in the P2+ lab, once we have a recombinant plasmid, then we use it to infect cells and then measure the functionality of TAT. However, over my research visits, I've been able to perform other techniques which are not routinely performed at HPP, such as co-immunoprecipitation, Western blots, and things like that, which I've been able to, which I then bring back to the lab and kind of implement in the lab at HPP. So as I previously mentioned, that I showed the genetic variation within TAT, as well as this alanine at position 21, in which patients had significantly lower viral loads than patients with the proline at the same position. So moreover, when we investigated its functionality, we found that this mutation had reduced functionality. So having this particular mutation at position 21 actually reduces dramatically TAT's ability to actually perform its function. And also on my last research visit, as I mentioned, which was in Europe, we were able to reveal the actual mechanism which is responsible for this reduced functionality. And that is involved with TAT-TAR binding and kind of a bit complex protein dynamics, which I won't get into. But the major highlight of this research visit was really revealing the mechanism for this reduced functionality, which we had previously observed. So what does this mean basically for HIV research, and particularly for HIV cure research? The findings from my study were able to provide a better understanding of the mechanisms which govern TAT functionality and HIV transcription, and also shed light on the dynamic interplay between TAT and this host transcription factor. The study also informs HIV cure studies, which is really, really important in us kind of collectively working together to finding a cure for HIV. So what we're hoping for, basically as scientists in the near future, is to reach what we call the 90-90-90 goals. And this is a global kind of goal in which we would like to have 90% of all people living with HIV knowing their status, 90% of those people living with HIV achieving antiretroviral therapy, and also 90% of those achieving this antiretroviral therapy having achieved viral suppression. I think all of these are really, really important in us also curbing the number of new infections that we see in our country as well, and really and getting this under control. And also, of course, in achieving the functional cure, if you remember, and that's having people infected with HIV off therapy, but still are virally suppressed. So their viral load still below the limit of detection. So in closing, I would like to acknowledge my supervisor. Oh, I also forgot to mention that the findings from my study are being prepared for publication. So very soon, we're going to be publishing a manuscript on those findings. So another thing I love about science is teamwork. As you notice, I've been saying throughout my presentation, we, sometimes I, but it's a lot of we. We really work together. And if you've read scientific articles before, you'll notice that there is very seldom one author. It's really a team effort in achieving really important scientific outcomes. So I'd like to acknowledge my supervisor, Dr. Paradise Madlala at HPP, as well as our group members, study participants, staff and students at HPP, Prof as the director of HPP, and then our collaborating labs, in which I have visited. The first one, the Art Lab at the University of California, San Francisco, and then the Mahmoudi Lab at Erasmus Medical Center, which is in Rotterdam in Netherlands. I'd also like to acknowledge all the funders, the NRF, UKZN, MRC, many funders which have funded the science, of course, as science doesn't happen if people don't have money. And these organizations have also funded myself as well. So that brings me to the end of my presentation. Thank you very much. Thank you for bearing with me with my flu. And I'm really open to any questions or comments that anybody has. Thank you.
Thank you very much, Zakithi. I'm a little bit challenged now. So basic science got hold of me. Without electricity, there's not much you can do. But thank you very much for this very inspiring talk. And if I can ask Sally to please handle the question and answer session. But before she's going to start, as a host, I like to have the first question. Zakithi, how much actually did COVID impacted on your, doing research, HIV research? I know that every, institution, everything just kind of suddenly rushed towards, COVID and the cure and whatnot. And then I think the HIV research somehow kind of, I had the impression always was kind of neglected a little bit.
Yeah, I agree. Well, I think the biggest effect personally that COVID had on myself as being a lab-based researcher was that we were not allowed to go to the lab. So COVID was really given preference. People that are doing research in COVID, and this was not only happening in South Africa. So for me, when the COVID pandemic actually started, I was in San Francisco on my first research visit. And there, the only people allowed to go to the lab were people that were doing research on COVID. So this was not to say that these other viruses, such as HIV, had now disappeared. They had kind of just been given preference. And also, as Dr. Sandile mentioned yesterday, that because a lot of funders were funding COVID research in the HIV research that they were doing at the time, they kind of inserted COVID into that as well. So it really, I agree with you there that the other viruses kind of took a back seat, and that really affected me personally and the progress of my PhD project. But I think that, so since then, since COVID has now, really highlighted the important role of scientists, I think people are really seeing, no matter what you're working on as a scientist, it's important. And not only that, because now we're in a pandemic, that everybody must focus on that. And there's still other things, which in my opinion, I think HIV is actually more devastating, has been more devastating than COVID, that we need to focus on.
Yeah, especially as if it shows that there is really currently no quick solution, still no quick solution, even after all these years of research. Sally, if you want to take over the question and answer session, please.
Thanks very much, Tanya, while you sit in the dark. Zakithi, there's several questions that have come in. I see we short on time, so I'm just going to read them out to you. Sure. Siyabonga wanted to know, is it possible to reproduce stem cells in the lab?
Unfortunately, I'm not involved in that kind of research, but I know that it is very difficult to do so. And that is why, you know, in these classic case studies where we've seen of patients being cured of HIV, it's really the stem cells which come from the donor. And I think stem cell research has really, you know, since then, since the Berlin patient was cured, has really been working hard in trying to reproduce that so that we can apply it. But I still think that, you know, it's not really going to be able to be applied on a large scale, because this means that everybody would have to undergo a stem cell transplant, which is a huge invasive operation. So even though we may get to a time where these cells can be reproduced in the lab, still how are we able to give infected people these very hectic operations? So to answer your question, I am not sure. In stem cell therapy, I doubt it that they are being able to be reproduced. But even if they are, we can't really apply it.
This leads maybe into the next question that was from Zamambo. So I'm paraphrasing what he said, but he says there are people who can't get HIV because of their mutated genes that they have. Mm-hmm. So why can't we artificially engineer their genes and use that to cure?
Yeah, so that's one of the strategies that I touched on briefly, which is the CRISPR-Cas. It's gene editing. So it's really, you know, artificially engineering these genes of infected people to maybe now express this mutation. So that is still also under a lot of work and research. And I think also we have a bit of work to do on that.
Okay. Then there was a question from Sitembile, who wanted your opinion on attenuated vaccines versus mRNA vaccines
in general. Well, that's a difficult one. I think that she's asking in relation to COVID. I'm not sure, or maybe influenza, where you do get an attenuated vaccine, but it really depends on the virus. You know, if you're getting an attenuated vaccine, is that going to really elicit your body or kick your body into action into making antibodies, or is the mRNA vaccine, you know, a better way in which the virus will respond? It really depends. It depends on the virus. So I can't really give my opinion in that sense. For me, it really depends on what works for that particular virus.
Okay. Then Siyabonga again wanted to know, will healthy eating help if you're not on antiretrovirals?
Sheesh, I know that a lot of people that are on antiretroviral therapy are still encouraged to eat healthy. I don't think that just eating healthy will manage the viral load of an infected person, because what the actual antiretroviral drugs are these drugs that target different parts of the life cycle of HIV, if you remember in my explanation. So we have particular drugs which target integration. So they will really try and stop the virus from integrating into the genome. Then we have another set of drugs which target the budding process, you know, and then other drugs target the assembly of the new virus that will bud off. So these are really specific, you know, when a person is on combination antiretroviral therapy that target the different parts of this viral life cycle in bringing down the viral load. So I can't really say that, well, I wouldn't say that healthy eating alone can manage the viral load of an infected person.
Okay. I was going to ask you how antiretrovirals work, so thanks for explaining that.
Sure.
And then here's a question that's coming again from Sitembile. How does PEP treatment, and it's disappeared, for HIV work?
Yeah. So with PEP, I'm not sure if she's talking about maybe the post-exposure prophylaxis, and we also have what's called PrEP, which is pre-exposure prophylaxis. So in pre-exposure prophylaxis that's taken by people that are HIV negative to prevent them from becoming infected. So that really allows a person that's uninfected to have a certain level of these antiretroviral drugs in their blood, that if they come into contact with the virus will not integrate and infect these healthy cells. But with PEP, which is post-exposure prophylaxis, it has to be taken within a particular time frame. So that's 72 hours, if I'm not mistaken, before the virus has really, caused havoc in the body, and that's in integrating. And then once the virus has integrated, it's very, very difficult to go back from then, because it's now like the sergeant in your body that's instructing all your cells to make new virus, spread, replicate, and this will replicate and infect other healthy cells. So that's how that works for treatment of HIV. I wouldn't say that the person who's taking PEP, they may have come into contact with HIV, but the virus is not yet integrated to be able to instruct the body to make new viruses to infect cells. So that's how that works.
Okay. Just a couple more questions. There's lots of people saying, thank you for such a wonderful talk. And then Lungasile wants to know, considering the nature of HIV, from some of the research, could there be any other tissues that get infected besides the hematological system?
Yeah, so that's the thing with this HIV, kind of really plays a game of hide and seek, and we are still seeking, all these reservoir sanctuary sites where the virus hides and really lies dormant and latent within these tissues. But HIV really infects a number of tissues within the body. It's within an infected person, I think there are very few tissues where you wouldn't find the virus. So it's really all over.
Okay. And then we're running out of time. So just one final question from Tafadzwa. Says, is there something called an HIV carrier that exists?
So this one is a very interesting topic. For myself, and being a scientist, I believe in science. So for everything that's said, I need to seek the evidence, which I haven't for this HIV carrier. I know there's been a couple of stories in the media, of people being said to supposedly carry the virus, but they are not infected by it. I mean, in this case, the only thing that I can think of are those people with the rare mutation that I described, that they actually do have virus in their body, but because the virus is unable to infect the cells, the virus, they don't have it integrated into their genome. So this doesn't mean that those people can't pass it on. For as far as I know, I stand to be corrected for that. But in terms of a HIV carrier in another way, I'm not aware of.
Okay. And then one final question here. Is there a role for indigenous and traditional medicine plants in fighting HIV?
Yes, I think this is a very exciting, especially for Black people, and Indigenous people who have believed in traditional medicine for centuries. There's actually an African traditional medicine lab set up in Howard, and I actually had the privilege of speaking to one of the heads of that lab, which is Prof. Neba, and they are investigating now in a scientific way, showing proof that particular plants play a role in managing viral loads of infected people. So definitely, to answer your question, yes, I do believe there is a role that traditional medicine plays in fighting HIV. And I think it's very, very important, especially in our country, where a lot of Black people are infected with HIV, and a lot of Black people do believe in indigenous or traditional medicine. So, yeah.
Okay, thanks. Zakithi. Tanya, over to you.
Okay, thank you so much, Sally, for handling the question and answer session. It's very difficult to read it on a small screen, and apologies for not putting up my video. You just have to live with my lovely Photoshop when I was still, like nicely dressed and made up, dolled up. Okay, so we are at the end of our National Science Week lecture, but not at the end of our public lecture series. What do scientists do? A look inside the ivory tower. So the lecture series continues, and we have other exciting talks lined up. And the next lecture on the series will be had at the 7th September, same time at 17:30. And the speaker will be Dr. Justin Pringle, and the title of his talk is why it's important to understand how fluids move to predict our future climate. But let me just thank Zakithi. It was really an amazing talk, and we had such a good response for people, and they're complimenting you. Fantastic. And even as a non-biologist, non-virologist, absolutely amazing. So I would also now like to take this opportunity to thank a few people. Of course, Ms. Mkhize for her inspiring talk. And it was really inspiring about her research and the passion she shows towards STEM education. And she speaks out of my heart that there is a lot of things to do, and we need young aspiring scientists, especially female scientists, that can take on mentorships and being a role model for future learners. To pursue careers in science. I also would like to again to thank Dr. Zakithi Mkhize, who presented yesterday on his fantastic talk about his basically the behind the scenes tour. Ruanne Peters, a very big thank you to you from the African Health Research Institute, who basically allowed us and some learners to visit the labs, the RE labs, and also who organized the speakers for tonight. And I think she did a very excellent job on this. Hannah Keel for helping us with the technology behind the scene. The researchers, lab technicians at RE for their help. I also would like to thank Sally, for taking over in this very interesting situation here. I'm sitting here with candles and a flashlight. Thank you, Sally, also for coordinating the event, for sending out the message to everybody. Ashok Raj, who designed the invitations and also who do the wonderful backgrounds. I don't know if you saw mine. The Department of Science and Innovation and SASTA for providing such a great platform to highlight science and engineering, and the countless people again behind the scene that you usually don't get to see, but they are very essential for the work that we are doing, and who also help to spread the message. Please remember to join us on the 7th of September at the same time, 17:30, for Dr. Pringle's talk. And I would like you to please unmute yourself if possible, and put your hands together for Ms. Zakithi Mkhize for her brilliant talk and inspiring talk. Thank you so much. And I really hope that
Thank you, everyone.
we can all sit again together and have live events, and not like this how shall I put it, technology driven events. And thank you so much, Zakithi, for giving us your time and preparing the talk. And I know you're a very busy woman, but thank you so much again.
Thank you so much. Thank you to everyone for joining as well.
All right, guys, this is it for tonight. Happy load shedding from my side. I'm sitting here till 8 o'clock. For all the other ones, enjoy the electricity while you still can. All right, good night, everybody. Cheers. Bye-bye.